Welcome to the Medicine channel. Let's continue the gene series topic replication. Time for disclaimer. Every replicating parent cell during cell division doubles its genetic material, the DNA, in such a manner that the daughter cells receives complete genetic material. Parent DNA strand serves as the template to be copied to form daughter DNA strands. This process of DNA formation by copying from a template DNA is called the replication. DNA thus formed is incorporated into daughter cells such that one strand is from the parent DNA and the other strand is newly formed daughter DNA. This is called semi-conservative replication. Let's move on to the steps of replication. Step 1 is initiation, the beginning of DNA formation. Step 2 is elongation, the continuation of DNA formation. Step 3 is termination, the end of DNA formation. Additional step 4 is post-replication modifications that happens thereafter. Let's drive through the direction of replication. Parent DNA has two strands with one strand running from 5' to 3' end and the other strand running from 3' to 5' end. The formation of new daughter DNA strand always starts from 5' end and ends with 3' end. Thus, DNA formation involves 3' to 5' read in direction, and so the direction of replication is always from 5' to 3' end. Let's see step 1, initiation. Along the stretch of the parent DNA strand, initiation sequence rich in 80 bases or a consensus sequence called ORE, origin recognition element. ORE is identified by enzyme protein complex remembered as TAMS. T for topoisomerase, A for DNA A protein, B for DNA B protein also called DNA helicase. SSB stands for single strand binding protein. This complex called ORC, origin recognition complex. DNA A protein melts the DNA strands at 80 rich initiation sequence. DNA B protein unwinds the DNA strand further, separating the strands ahead. This creates overwinding at the ends of DNA strands called the DNA supercoils, and the enzyme topoisomerase relieves these DNA supercoils. Single strand binding proteins binds to the unwinded single strands of DNA, preventing from rewinding. This creates a V-shaped separation called the replication fork. The nuclear fluid in the region bubbles at the separation, creating a replication bubble. Formation of DNA needs a short stretch of RNA called RNA primer. Primer formed by the enzyme primase, also called DNA G protein. While in eukaryotes, primer formation is done by DNA polymerase alpha. Once primer has been formed, new DNA strand synthesis is initiated by the enzyme DNA polymerase 3. This is initiation of replication. Let's move on to step 2, elongation. Following DNA unwinding and primer synthesis, DNA polymerase continues a sequential addition of deoxyribonucleotides complementary to the bases on the parent DNA strand following base pairing rule. This happens in both the strands, and so the process of replication is bidirectional. Adenine on parent strand specifies thymine. Thymine on parent strand specifies adenine addition. Guanine on parent strand specifies cytosine addition. And cytosine on parent strand specifies guanine addition. DNA polymerase 3 does this action in prokaryotes, while DNA polymerase epsilon and DNA polymerase delta is responsible in eukaryotes. Upon further unwinding, elongation continues in both the strands such that continuous strand synthesis occurs in one strand called the leading strand. Discontinuous strand synthesis occurs in the other strand called the lagging strand, also called the Okazaki fragments, making replication a semi-discontinuous process. While DNA polymerase III synthesizes both leading and lagging strands in prokaryotes, DNA polymerase epsilon synthesizes leading strand and DNA polymerase delta synthesizes lagging strand in eukaryotes. Then RNA primers removed and replaced with DNA by the enzyme DNA polymerase I. The gaps remaining were sealed by the enzyme DNA ligase in prokaryotes, while ligation in eukaryotes is done by DNA polymerase beta. And this is elongation of replication. Let's end with step 3, termination. Following elongation, replication forks from both the ends collide over a region. This terminus region contains TER termination sequence. They are recognized by terminus utilization substance, the TUS proteins. TUS TER complex arrests the fork and thus terminates replication. And that concludes replication. Ending on a social note, wheels on road are the worst killers in the world. Life on earth is more beautiful than life under wheels. Every being reserves their right to live, so always drive slow and ride safe. And signing off for now.